All right. Hello, everyone. It's 7 o'clock. And so I'm going to go ahead and get us started. I'm sure we'll have a couple more people straggle in. But my name is Anne-Marie. For those you who have met me, Anne-Marie Reed, and I'm the program coordinator for Travis Audubon. We're very excited to have Dr. Pamela Rasmussen here. Before we get started, I have a few announcements. Mark your calendars. Birdathon opens on March 3rd. We've got a lot of really awesome trips coming up, so keep your eyes open. The Katura Canyon Valentine's Appeal is happening now. We also have our spring class schedule, so go ahead and check that out. And a few things before we get started. If you could all please turn off your microphones and mute. We do love questions. If you put any questions you have in the chat box, we'll include them in our Q&A session at the end. And we do have a QR code on the screen. And if y'all wouldn't mind taking a few minutes just to answer a quick survey, it helps us know who's attending and just gives us a little information. But now I'm going to pass it over to Suzanne Coe. She's one of our board members, and she's going to introduce our speaker tonight. Thank you all so very much for being here.
Hi, everyone. Welcome. It's great to be here, and I'm honored to introduce our speaker this evening, Dr. Pam Rasmussen. Pam grew up birding in Oregon, and after receiving her PhD, worked at the Smithsonian Institution before joining Michigan State University, where she taught biology, including ornithology. As a teacher, she has led study abroad programs to Uganda, Borneo, Indonesia, Antarctica, and Costa Rica, among other places. And during her world travels, she submitted eBird lists for more than 70 countries. Today, she's the lead taxonomist at the esteemed Cornell Lab of Ornithology and updates the Clements checklist that underlies eBird, Birds of the World, Merlin, and other lab projects. She also serves on the NACC and SACC, the North and South American checklists, the IOU's working group Avian Checklists, WGAC, now Avalist, which is aligning global avian checklists. She also co-authored Birds of South Asia, the Ripley Guide, and has co-described 11 new Asian bird species to science. This evening, she's going to give us an overview of recent taxonomic updates and discuss the importance of avian taxonomy to citizen science and how important citizen science has become to avian taxonomy. That word taxonomy is a real tongue twister, and I'm glad I don't have to say it anymore. And I will now turn things over to Pam. Thank you for being with us today.
Well, thank you very much. And I hope I don't have the same trouble with the word taxonomy. So, thank you for inviting me. I'm glad to be here, and well, here being Michigan, but glad to be speaking with you tonight. Let's put it that way. And anyway, so my topic today, as Suzanne mentioned, is avian taxonomy in the era of citizen science. And what do I mean by that? Well, basically, I hope to show how avian taxonomy benefits from citizen science and how all of us can have an impact, a positive impact on avian taxonomy. And also, conversely, how citizen science benefits from avian taxonomy. So how does it benefit the user, the birder, et cetera? Well, as we'll see, that's largely by organization and updating of data. We're also going to talk about some of the updates, the taxonomic updates that happened recently and some that are projected to happen, and also talk a bit about the checklist alignment project. So it'll be a bit of this and a bit of that, and I'll be happy to answer questions if I didn't cover the area that you are most interested in or particular questions. So first, what is taxonomy? I think everyone knows, but best to make sure. Basically, taxonomy is the classification of organisms. And organisms, if it is described as science, it is a taxon, plural taxa, of course. So that's not just species, that's also subspecies and orders and families, et cetera. All of those are taxa at different levels. But we're going to talk mainly about species limits today, and I'll explain why. And the fact is that most of the taxonomy of birds that has been done in the past was based entirely on their external morphology. And that's because that's what the ornithologists who were doing taxonomy had to work with. And now, ideally, we use integrative taxonomy. We use whatever pertinent, relevant characteristics we have data on that we can reasonably bring to bear to ascertain whether they are a species or not. And that obviously includes genetics, includes morphology, which is still as important as ever, but it's not the only character that we use now, or only character set, I should say. We also use vocalizations increasingly, and whatever we know about reproductive isolation. And it's a fact that most people are most interested, most birders, I should say, and ornithologists are most interested in species limits. So that's the taxon level of special interest. And here's an example that you may have noticed changed this year, the herring gull. And that would be that if you if you don't travel out of the US, you have one species still probably, unless you manage to get a Vega gull somewhere. But if you do travel out of the US, you might have 4. And for a long time, it was recognized as 2, 3, or 4 by some lists, but not by all, including eBird, Clements, and North American Checklist Committee. But now eBird anyway recognizes 4 species. And I've put the juveniles here because the differences are most pronounced in juveniles. These two look virtually identical, but they're not even closely, well, they're all closely related. All these big gulls are closely related, and they would all probably hybridize given a chance. But these are not each other's closest relatives, let's put it that way. And they're not vocally identical either. So they're similar, but there are some pretty consistent differences, vocal differences. And then when you get to Asia, well, the Vega gull is quite similar vocally to these two, but it's not that closely related either. And it's also readily distinguishable. And then the Mongolian gull, which has been lumped by some and not by others, and sometimes lumped with the yellow-legged gull or Larus cachinens. I shouldn't say yellow-legged, but that group of yellow-legged gulls is a steppe breeder, and its vocalizations are quite distinctive. And it also is not the closest relative of Vega gull, although these relationships are somewhat tentative and remain to be fully worked out. Anyway, so this is one example of a major split that happened this year that affects your life list if you travel much. So, excuse me, so why are species limits of special interest? Well, for one thing, they're the units that are most used in scientific study. They're the units that are most used in planning for conservation and in action. And in order to do that, the importance of various bird areas has to be assessed, and the status of the species that are affected has to be assessed by BirdLife. And so all this goes into the assessment and planning for conservation. In addition, law enforcement generally uses the species limits, the species level as the unit that they deal with. And so if something is considered a subspecies, it may not be protected in some places, like some of the starlings that are in the trade in Southeast Asia. If they're considered subspecies, they're not protected, but if they're considered species, they might suddenly be critically endangered and protected. And then, of course, there's birding. And I don't know what proportion of you consider yourselves birders more than ornithologists, but many of you probably do, and it's important to you, right? It's important to me. And here's an example of how what was a fairly rare bird found on 2 islands in the Lesser Antilles, excuse me, the white-breasted thrasher, with further study of its vocalizations and its genetics and its plumage morphology, has been shown to be better considered to be two separate species And now each of them is restricted to really small areas. This Martinique thrasher is only found on one little patch of land in eastern Martinique. And this particular individual comes out into the parking lot and begs for scraps. But anyway, they're quite rare. They're of great conservation concern, and being listed at the species level does provide them that at least increased level of interest and protection. Not that that should be a reason to consider them a separate species, but it has that impact. The St. Lucia thrasher is not quite as rare, but it's also extremely endangered by the fact that its habitat is quite restricted and subject to continuing development. So what is a species? How do we define species? Well, we use two major species concepts. There are many. There are probably over 100 different species concepts that have been published, but in ornithology and most aspects of biology, we use two different species concepts. The PSC, or phylogenetic species concept, in which a terminal branch on a tree that has members sharing diagnostic characters that they obtained through common descent is a PSC species. A BSC species is a group of interbreeding organisms that are reproductively isolated from any other such groups. Well, in practice, many of taxa are species, whether you consider them a PSC species or a BSC species. But also in practice, there's going to be many more in the PSC than in the BSC. PSC does not recognize interbreeding as really relevant, and generally, but not always, does not recognize subspecies in the PSC. But the fact is that almost all lists that have been generated and widely used, and all of the major global taxonomic lists use the BSC and always have. And that's because there's a lot of problems with the PSC, such as somewhat arbitrariness and what exactly does it mean that something is reciprocally monophyletic, and how many diagnostic characters do you need? Like, do you need just one genetic mutation to call something a species, etc. So there's all kinds of problems with the PSC, and it has not proved to be very workable and has not been adopted by the major committees. In general, the BSC that is now followed, though, is not really the BSC of Ernst Mayr, who was influential in causing the lumping of many groups of birds into very, very polytypic species that really didn't matter what they looked like. As long as it was considered possible that they might interbreed, they were lumped into a single species. Now we use an approach that is more what we call the modified evolutionary lineage approach, where we don't assume that they're going to be able to interbreed and don't consider them to be the same species if there's just a small amount of known interbreeding, for example. Okay, well, again, most species were originally described based solely on their morphology, and that's because that's what ornithologists had. And typically in the earlier days, a population that differed consistently in multiple ways from any others was considered a species. But if there was only one or two differences, when the subspecies concept was enacted, which was early in the 20th century, subspecies were characterized generally by just minor differences, like a shade of gray or a difference in size, et cetera, or just a few differences. But we have some problematic cases. We have flickers. Flickers, for example, are among those birds that just don't seem to see each other the way we do, and don't discriminate on the basis of what we would think are species level differences. So we have flickers that of pure phenotype that are very different in the Pacific Northwest, et cetera, from the flickers that we have here in Michigan. but then in the Great Plains, we've got everything in between over a very wide area. These birds are not looking at each other and saying, oh, that's, that's not acceptable. It's fine with them. So flickers are kind of an exception, but not the only one. And nevertheless, we don't assume that just because flickers don't discriminate on the basis of plumage, that doesn't mean that that applies to all woodpeckers. We also know that many birds have UV vision and coloration and ability to see much more detail in terms of color than we do. They have more cone types than we do, and many of the cone types are doubled instead of single. So they do have better color vision, most of them, and they do have the ability to see colors that we don't see. So for example, these mountain tanagers from South America have been shown to have bright color patches that differ from what we see. So they're emphasizing, based on the UV coloration, these color patches. They're really not noticeable to us. So that's just an example. And there's so much more to be learned about all this, but we do know that birds don't see each other exactly as we do by any means. And we also know that birds use their vocalizations in ways similar, but not identical to ways that we use them. And some of them, really, that's all they've got. The willow and alder flycatcher are famous examples. They were long thought to be the same species. They look essentially identical. You normally can't tell them apart unless you can get measurements, and even then, maybe not. But their vocalizations are quite different, and they know it, and we know it if we can hear them. So they don't need to be able to tell each other apart visually. We now have a much improved ability to deal with vocalizations, to obtain vocalizations and to compare them. In the past, it was really quite challenging. You had to have highly specialized and very bulky equipment, very expensive, and you had to go to a lot of trouble. And it was all on tapes or recordings. Records, etc. You had to have special machines to get sonograms, etc. Now it's something you can do so easily that the archives, publicly available archives for vocalizations, have just taken off and are an incredible resource that we'll talk about more. Anyway, so now that we can do this so much more easily, well, now we have a profusion of data that often is too vast to work with easily. But in many cases, that's, we have the ability to work with species concepts that are complexes that have been known for a long time to require taxonomic action, but it hasn't happened until now because of the fact that it was just so much more difficult before. And we also are able to often find that there are new species out there that weren't recognized before based on their vocalizations. And a good example is the description of several new species of Amphitta in the Rufous Amphitta complex a few years ago. Finally possible based on the availability of recordings of each of the taxa coupled with genetics and etc. Anyway, but we still have obstacles. For example, often we have very few or even no recordings, or poor quality recordings, or recordings that don't indicate what the context was, or maybe not easy to interpret whether recordings that may be different are actually homologous or not. We also have the problem that some birds make it difficult because their vocalizations are not straightforward. Some of them learn their vocalizations to some extent, and it's difficult to know which of those possibly learned vocalizations actually have taxonomic information. And in addition, of course, there are plenty of birds that have dialects as well. So it's difficult to know whether regional variation may be taxonomically informative or maybe just be dialect. And then, of course, there's taxonomic inertia, meaning that we have, well, taxonomy is challenging and takes time and isn't very well rewarded generally. And And so we have inertia. We have papers that people have been working on for years. I'm not the only, I'm one of many that has that problem. Papers in the, a backlog of papers to work on. And you always think you're going to finish it in the next year or so, but and then just the fact that even when we get papers finished, they may be not as conclusive as a taxonomic committee may wish for in order to make a decision. So taxonomic inertia has various causes as well. And there are quite a few cases, some of which we will talk about more, like the herring gull case, where we've known for a long time that the present taxonomy isn't adequate, but we haven't known exactly how to deal with it yet. And we also know that birds don't hear sounds exactly as we do. Their inner ear structure, middle and inner ear structure, is quite different from that of mammals. And it's quite good, though. They have excellent hearing. And some of them actually appear to have much better discrimination of sounds, like very, very rapid paced sounds. They're able to discriminate much better than we can. But very little study has been done on this. And so we don't really know how broad this is among birds, or how many birds have that ability. But we also know they have abilities that we can't even comprehend, like distinguishing each other by voice. Like emperor penguins can distinguish their young throughout the period of development. And king penguins can come back to their colony after being away for a few months and seek out their one offspring based on its vocalizations in a colony of hundreds of thousands of other offspring that look identical. So they have abilities that we can't even, haven't figured out yet. But anyway, so nevertheless, we can use their vocalizations to a great extent in taxonomy. Here's an example of taxonomic inertia. We've known for a long time that great egrets of the old world, generally, when they're taking off, at least when they're taking off into flight, they give a rattle. We've known that the American great egret, gives a croak. And the croak is basically a much faster rattle, but it's so fast that it sounds like a croak. Anyway, but the thing is that it's not so easy to deal with some of the other aspects, like the fact that the archives of recordings don't necessarily give context, and so you don't know whether you're dealing with birds at a colony that are giving a completely non-homologous type of call, etc. And in addition, these birds have very transient colors in the breeding season that distinguish them very well, but then the rest of the year they're not so different looking. Anyway, so that's an example of something that we know we need to fix, but hasn't been done yet. Let's hope in the future pretty soon. Anyway, so prior to the advent of citizen science, taxonomy was the specialty of taxonomists. There weren't very many, and they were mostly museum authorities who specialized in a particular group or in birds of a particular region, etc. And they worked with museum specimens. And that is how they were able to do taxonomy, because there was nothing else that you could use. And our very important works like Birds of the World by Peters, the Peters series that started out being written by Peters, and then when he died was taken over by others. This is an extremely important work based on museum collections, could only have been carried out in museums. And nevertheless, this of course has its disadvantages, one of them being that the decisions were made by an authority, and there was no dissent, and there was often no justification published. It was just done by fiat. Well, now we have citizen science, obviously, and we have three major ones that I'm going to talk about briefly, mainly eBird. So eBird wasn't started all that long ago, just 23 years ago, went global in 2010, and I didn't get started until 2014, and immediately became an addict. But anyway, so eBird has exploded. It has become an incredible resource, and I would say that even if I didn't work at Cornell, I'm not a part of the eBird team exactly. I'm the taxonomist, so it's related, but anyway. So there's so many features. I don't know how many of you use eBird, but so many features that just make it amazing, and it's just getting better all the time. These are some of my favorites. Bar charts, whenever you're about to go somewhere, are incredibly useful. The quizzes, the ability to search photos, sounds, etc., customize your search, and the ease of finding species, just finding information instantaneously about where a species occurs. I use it like many times every day in my work, and I'm sure many of you do as well. So what does eBird offer you? Well, of course, those customized lists that when you're entering your data that give you what you can expect that day in that place, rather than an entire list of the birds of the state or a list of the birds of the world, etc. You have a customized list. And you can use these bar charts to figure out what's likely, like where this was somewhere in New Zealand, where I was recently, where the white-capped albatross was one of 3 main albatrosses that you could expect there, but certainly not bullers or salvins in that particular area. And then quizzes are really helpful for getting ready to go somewhere. Also, the targets for at the species level anyway tell you what you haven't seen in the area. Of course, rare bird alerts that can be customized in various ways. But things that are especially good about eBird also include the possibility of fixing your records if a problem is found, a misidentification is found, or let's say you made some recordings and you didn't get to check them until a year later, but you can still fix your list if you find that you had a species that you didn't have in your list. You can add it in later. You can also back up and provide evidence using photographs, videos, recordings, etc., which is really a really great way to make sure that people know you really saw that bird, or if there's a mistake, it can be fixed. And also it provides a great way to organize all your stuff. So now you can just search on a particular species and see what photos you have of that species and where you saw it, when, etc. Oops. And then also language prefs. You can set the languages for both eBird and Merlin, etc., to your preference. And of course, you get your automated taxonomic and nomenclatural updates and so much more. So I feel like there's a lot more that I don't even know about, but those are the main things that I use a lot. What do you offer eBird? Well, assuming you're a birder, your checklists and your media. So it all goes in to make this big picture, which is to document occurrence and many other aspects of birds of the world. And even record shots that maybe aren't that good. I mean, of course, great shots are better, but record shots, as long as they're identifiable, are useful as well, especially where you've got rarities or difficult IDs. So it makes all the difference if you've got that record shot, if it can be checked later. And of course, if you are a frequent eBirder, then you get to a point where you're able to report apparent misidentifications. If you find, and there are lots of them. We know this. It's a constant struggle for the quality control people. So there are lots and lots of reviewers who are on this all the time, but something slipped through, or they're too busy, or they're traveling or something. And there are some areas that don't have the reviewers that are needed, but it's getting better all the time. Anyway, so and then also the ability to rate photos, so the good ones will rise to the top. Okay, here's a couple of examples, maybe embarrassing for me personally, but I'll just do them anyway. So I was in China not that long ago, and I really wanted to see Mongolian gull because I'm interested in this whole herring gull split, and I hadn't seen it before to my knowledge. So I went to a particular area and got some photos of Mongolian gulls. Well, when I came back, it turned out that this one is definitely a Mongolian gull, but we don't know what this one is. Is it a Vega gull or is it a Mongolian gull? If anyone can tell me, I'd be very happy. But so far, I've had some experts on the case, and we're still stuck at Vega/Mongolian gulls. So even though they're probably not each other's closest relatives, and they're distinct ecologically, and they sound different, I can't tell that from these photos. And nobody else seems to be able to either. So they're in purgatory right now. And I'd like to get them out of there, but anyway, it's okay. And then here's another example. So this photo, at least 10 people have rated it, and it's still at a terrible. So why am I showing you this such an embarrassing thing? Because I struggled and struggled to finally see this bird. And it was getting dark, and it was across a ravine. And we saw them fly into the tree, and I just snapped some shots with no hope of actually getting anything. And then when I got back to my room, I found this little head popped up. So this is what I mean by a record shot. It's really bad, but there's an imperial Amazon in there. And I've got proof, right? So I'm not proud of it, but it's still better than nothing, right? So anyway, yeah, good shots are great, but record shots are okay too. Okay, so on to a couple of other citizen science projects that probably some of you use, including iNaturalist. I don't personally use it much, so I can't say much about it, but it is incredibly popular, and of course it goes way beyond birds, but it's got a lot of people contributing bird data as well, so it's also very important. But it's not nearly as user-friendly in a lot of ways in terms of the output, in terms of what you can get out of it as eBird is. So it's very, very different in a lot of ways. And then Xenocanto actually is incredibly useful, and I use it all the time too, but it's only sound. So it has some aspects that are really great. For example, prior to the split of common chaffinch, here's what the range looked like. They hadn't adopted it yet. They're a bit behind on that because they're busy adding all these other taxa in, like mammals and insects. But anyway, so one of the things you can do is you can just click on the race that you are interested in. So you can just instead of having to sort through and find all the ones from Madeira, you can just click on the Madeira race, whichever one that is. Madeirensis. Anyway, and then you've got the Madeira ones. So really handy in a lot of ways and useful. There are lots of people that contribute to Xenocanto that don't contribute to Macaulay Library, which is what you do when you put a recording in eBird. And then there are some that do the same recording to both. So sometimes they're duplicates. Okay, so now what about the major taxonomic committees? Well, we've got 4. So there's the Howard and Moore 4th edition by Dickinson and Christitis, and Dickinson and Remsen. And that was last updated in 2013 and 14. And that's the 4th edition. It's online, and it's very, very useful. I use it all the time. But it's not actively being updated online anyway. And then we have 3 others that are widely used. The IOC World Bird List. I'm one of the managing editors of that, but I will say David Donsker has done the lion's share of the work on that by a long shot. And that was started by Frank Gill, who's still involved. And then eBird Clements, which I'm now involved with as well. The Clements list. It's the same list, but it's different versions of it are used for eBird versus for Birds of the World, etc. And then the HBW, or BirdLife International checklist, which is on BirdLife Data Zone, and it was published. It is a modified version of what came out of the checklist from 2014 and 16. That is one of the most useful books of all time, in my opinion. which enacted a lot of splits, hundreds of splits based on the 7-point scoring system that has been so controversial, but also useful in some ways to point out many cases where splits really are needed. Anyway, so we got these 3 lists, and we've all gotten together, these 3, not Howard and Moore, but IOC, Clements, and HBW, BirdLife, to consolidate, because it is a problem to have multiple checklists. As you know, you've got different names, you've got different taxonomies, and it is just, I mean, in some ways it's good because it reflects taxonomic uncertainty, et cetera, and individual decisions, but in other ways it's very confusing, especially where we've got multiple names for the same thing. And many people just find it confusing. Anyway, so we've the IOU, International Ornithologists Union, started a working group on avian checklist called WGAC. And we're now calling this AVILIST now that it's about ready to come out. And this is a species level aligned checklist for these 3. And it will be coming out in the next few months, probably Mayish. And and then there'll be updates. So the first version will be version 1, obviously, and then the idea is to update it every year. So we've already enacted many of the changes, the species level changes, and some others like genus level, et cetera, in the Clements updates and the IOC updates. So there's not that many decisions that have already been made that aren't already publicly available in these 2 sources. BirdLife is also aligning, but it takes them a longer time because they have many other aspects that they have to deal with downstream. So anyways, what's AVILIST? Well, basically, it will be this single global checklist for bird taxa. Of course, there could be an update to Howard and Moore. There could be another checklist that comes along. But anyway, we have AVILIST that will be these 3 aligned instead of having the 3 separate ones. And anyway, so we have voted on all of the species level incongruences and come to at least a majority vote. And in most cases, we had unanimity among the voters. But of course, everything, there are many cases where they're controversial, or we just don't have enough information. In many cases, we thought, well, these are probably separate species, but we don't have enough information to make that decision. But we did make a lot of changes. So we are using the IOC World Bird List 15.1 as a starting point. And that's the subspecies level taxonomy as well as the species level taxonomy. But again, it's almost all aligned. We haven't done subspecies yet, so that's still to come. We have a companion paper that is intended to introduce the list. It's not published yet, but it is out for review. And so we will not be having continuation of the Clements and IOC lists into the future. Exactly when those will cease is not clear, but it's in the next short period of time. And then BirdLife will continue to maintain its own list as needed, but it will be increasingly aligned as time goes on. So why do we need it? Well, to minimize confusion, because confusion leads to inaccuracies of data entry and analyses. And also, we also are trying to just make it consistent from one taxonomy, or one consistent taxonomy as to how species are treated. And nevertheless, we have many areas of uncertainty. Most cases do involve some uncertainty. And so we have summary statements that indicate where those areas are. And we need to keep our taxonomy updated based on the latest research and the resources such as the sound recordings, et cetera, that are now available. AVILIST provides a platform for other types of relevant taxonomic data as well. We have many fields in AVILIST, some of them populated from AVIBase, for example, that include the protonym or original name, the author of the name and the date, the reference for the description, the type locality, and many other fields as well. So this is all, many of it comes or is like similar to what's in Peters, but of course more up to date. And anyway, so huge effort with many people involved. And anyway, one of the problems, of course, with having multiple lists is that there's a lot of duplication of effort. And so this is also going to be avoided by having a single list. So what's next? Well, we have phase 2, which will be the annual update, and that'll include newly published taxonomic work and already published taxonomic work that hasn't been evaluated yet because it wasn't a taxonomic incongruence at the species level. And we have hundreds and hundreds of subspecies level alignment issues to deal with as well. We'll also have regional committees. So there's going to be committees for every major region that are in charge of coming up with proposals for changes. So how do taxonomists use the photo archives that we now have? Well, for example, we can use photos to make indirect comparisons. We have to remember all the time that there are lots of shortcomings with comparing photos, such as what the lighting was like and whether they've been edited and processed, etc. And of course, angles and there's all kinds of pitfalls, but they're incredibly useful. So they're useful in all kinds of ways. We just have to bear in mind those issues. And we can also often look at because we have so much data now for many species, we can also look at where we might have zones of intergradation, where we might find a lot of hybrids. And for example, in this case, where we recently split the Eurasian goldfinch, we found that, yes, we know there are some hybrids. This has been known for a long time, but the vast majority of birds from the largest, most of the range are certainly not hybrids, or at least not obviously hybrids. So we can really use this to zero in on areas that tell us that kind of information rather than having to use solely museum collections as in the past. And here's an example, the mealy parrot. So there have been 2 studies that show quite deep mtDNA divergence between mealy parrots from Central America and Mexico versus South America. And they look quite different. They had patterns are different, coloration's a bit different. So they really are, they seem like they should be separate species. And BirdLife International did split them, as did IOC. But the vocal analysis showed them to be very similar. It's possible that might not be a killer in terms of this, whether they're species or not. But what we found is that over a huge area, mainly Panama, but Panama is a long country, there seems to be intermediacy. You cannot classify them into one or the other. There's so many intermediates. So this proposed split really didn't make the grade. It's possible that it should, but further study is needed. We need to figure out what this apparent intermediacy or area of apparent intergradation is all about. And so further study is needed before these can be considered separate biological species. If it was just a really narrow area of overlap with a few hybrids, then there probably wouldn't be a problem. But with all of Panama basically being populated by intermediates, that's a problem. Well, what about museum specimens? So I'm actually a museum person. I've worked at museums for years and years, and I've done most of my work with museums specimens, but I don't think that museum specimens are any less important than they ever have been, based on the profusion of data we now have online. We still need museum specimens as much as ever. They're complementary. They hold the specimens that our knowledge of biodiversity is based on, including the name-bearing types, voucher specimen series that allow you to do direct comparisons from different localities under controlled conditions and don't have the same pitfalls that photos have. And also sources for DNA, isotope studies, et cetera, as well as the material that's needed in order to produce illustrations that are comparable. If you just use photos for illustrations, you end up with lots of problems that you can avoid if you have specimens sent to you if you're an artist. Anyway, so I would say that museum specimens are more valuable now than ever as our natural habitats and bird populations are reduced and in some cases lost, that specimens are irreplaceable. Here's an example, though, where the specimens tell us one thing and photos may show something different. And we have this case where HBW and BirdLife split the Cathvarius group of crimson-breasted woodpecker from the Pernii group, which is mainly Chinese and northern Southeast Asian. And one of the things that gave us a concern when we were evaluating that for whether we should adopt that split or not was that one of the subspecies that occurs in Tibet was classified earlier by the original describer in the wrong racial group. And that didn't make any sense until I saw that if you preserve a Pernii like this, it looks like the red is at the back of the nape, like it is in Cathvarius, but it actually isn't, because this is where it is in Pernii. That's one of the major differences. And it's just that you can stretch the skin and position it in such a way that it's misleading. And that must be what happened with that subspecies. So we see in photos, we can see that clear difference that can be obscured in museum specimens. We also, of course, have the problem that museum specimens can have, some of the colors can be transient, including like iris color and bill color. And I see I'm already running out of time. So is that a problem? Should I try to, Suzanne? Well,
sorry, I couldn't find my unmute button. I think you're
okay.
good for now. We did want to start questions soon.
Okay, I'll maybe skip a few things. I think I've talked too much, so sorry about that. Anyway, so in this case, we have these two scimitar babblers that actually were, their range was broken up by a different species, and nevertheless, they were still considered the same species because their plumage looks pretty similar. But when you actually get a series of photos, what you find is that their iris color and their bill color is really quite different consistently. And also the vocalizations show that the female component of this duet is quite different consistently. So now that we have these archives, we're able to make use of them to supplement museum specimens to make decisions. Okay, so how do we use sound recordings? Well, if we have stereotyped vocalizations, then voice is key, but we have a lot of issues with using them. Here's an example of a case where recently the brush cuckoos were split based on a vocal analysis by Wu et al. And when you hear these birds in the field, you recognize right away they're quite different. But of course, it takes more than just being able to say, oh yeah, they're different. You have to actually do the vocal analysis, and that can be quite complex. And you have to have enough data in order to do the analysis with. Here's an example of a case where back when I was doing the first edition of Birds of South Asia, I realized that these Himalayan woodpeckers were quite different than the South Indian ones vocally. And Paul Holt, who had recorded them, also had previously known that. And they didn't even respond to his playback. And so when I got a larger sample in our second edition, we split them. But now there's a paper that has formally analyzed those vocalizations, including the drum rolls, and found them to be quite divergent, much more so than many other woodpeckers in that group. But of course, things are more complicated when you find that there are individuals now known from an intermediate area, but nobody knows which one they are because they haven't been sound recorded as far as I know. So more work needs to be done. Anyway, so how do taxonomists use citizen science video? Well, here's an example where I could very quickly show you that if you're in Hispaniola, the palm crow has this elaborate call behavior of cocking its tail and drooping its wings really quite spectacularly. And the Cuban one doesn't seem to do that, but there's lots of video showing it in the Hispaniolan one, none in the Cuban one, although, I mean, there's one that shows it maybe a little bit, but it's not like it is in the Hispaniolan one. But there's also not very many videos for Cuba. So if you're going to Cuba, maybe make some more pro videos and we'll sort this out. They've already been split, but it would still be great to figure this out. And how is taxonomy used by citizen science? Well, of course, by organizing the data, providing the information about evolution and divergence and what level they're at, and also incentivizing. So, of course, we all, well, many of us keep species lists, keep photo lists, keep audio lists, I should say, life lists, and many other kinds of lists. And all that needs to be updated based on the latest accepted science. Well, I think I'm going to skip these because I did have these in the October update, and it's online as well. So basically, one thing I do want to mention, though, is that we do plan to have individualized reports for what changed in your life list when the taxonomy comes out the next time. I believe that that has already been, they've already figured out how to do it, and it's so it should happen. So that's something that will be really helpful for people who have, especially if you have a large life list, it can be quite challenging to know what exactly changed, what you lost and what you gained. Just quickly, the most taxonomic changes this year happened in Melanesia and Indonesia, fewer in the Northern Hemisphere and Australia. And these are some other types of changes we had as well. So quite a few generic changes, a few nomenclatural changes for various reasons, quite a few subspecies changes. We have many, many more of these that are upcoming, but they all have to be worked out and carefully considered. Okay, so how does it change? How does it affect you? Well, of course, when taxonomic changes happen, names have to change in many cases, ranges change, identification criteria change, status changes, etc. You're going to lose or gain species, hopefully gain mostly, but there are some lumps too. So it also has the result sometimes of leading to unidentifiable records, like those Corey shearwaters that you had off North Carolina. So often they end up in a bin that if you go in and you have photos, you can fix them, but many of them are just simply not going to be identifiable. Here's an example of one of the splits that affects a lot of people. And this is interesting because we knew for a long time, there are lots of studies now like this one that show we've got 3 main clades of barn owls, and that the Furcata clade is more closely related to this is the American one, and the Western European African one is more closely related. These two are more closely related than the Eastern one. But what seems to have escaped most people's notice is that the American one has quite a distinctive vocalization that is lacking in both of the Old World ones. So once that has been established, that really sealed the deal. What we also know is that this isn't the end of the story, because there's other taxa in this group that are probably species level, but at least it's a big step in the right direction. We also were finally able to deal with the island thrush complex. It is a monophyletic group, and so you could say, well, maybe it is all one species, even though they look completely different from one island to the next, or even sometimes from one mountaintop to the next on the same island. But we also know that they're vocally quite different, and they're different in it's clear that they're, to me anyway, and to all the members of WGAC, that we're dealing with multiple species. And we ended up with a 17-way split, which is not the end of the story. There are some other cases where splits are probably justified, but more evidence is needed. And many others. But here's one case where a citizen scientist, a bird tour leader, actually managed to get video and photos of a mystery bird that had not been recognized for many years because it was thought to be a hybrid or an abnormal bird, but in fact it is really quite distinctive, and it's incredibly rare, but he just happened to be at the right place in the right time and got video and got photos. So that finally takes care of that, and hopefully more people will find it. And then we had another new species come out last year, the Timor nightjar, which had been known for many, many years to be a new species, even though Timor is quite well known and it's quite common. Vocally, it was just too different to be the same species. But finally, the paper came out. It just took, it's a great paper with a lot of data, but it took many, many years for that to come to fruition. All right. So just very quickly, a little preview of some things that are going to happen most likely. One is that we're going to have 3 species of rockhopper penguin instead of 2. You've already got this one if you've been to Tristan Island, which very few people have. I haven't. So, but then these 2 distributed on opposite sides of the subantarctic are also quite different in some ways, but closer to each other than this one, which is clearly a separate species. So this is a kind of arguable split. It could go one way or the other, but the evidence, in my opinion, and in the committee's opinion, favors species status. Here's one where it didn't go that way, and this is the Campbell albatrosses. These are my photos from New Zealand recently. Campbell albatross mainly differs in having a bright yellow eye, but also a few differences in wing pattern, etc. And it breeds mainly on one headland of one island in Campbell Islands. And so some have accepted it as a separate species, but genetically, they're not more distinct than the 2 main populations of black-browed albatross. And black-browed albatross do land sometimes on, here's the island with the Campbell albatrosses. I took this recently. It's a fantastic place, just thousands and thousands of Campbell Island albatrosses, but a few black-broweds with black eyes find their way there, and they just hybridize indiscriminately. So the case is not really great for species status for Campbell. I kind of think they should be a separate species, but I can't really make a strong argument for it. Here's some more likely splits. The white tern 3-way split, maybe a 2-way split of the coppery-tailed trogon, and then the red grouse. And here's a lump. Nobody's going to want to hear this, but well, it's really hard to get some of these. So this one's the Macquarie one that I recently photographed, but these shags, really, there's not a good case for keeping them separate. They're extremely close genetically, despite the fact that they occur so far apart from each other and they're not pelagic birds. Somehow they've managed to colonize those disparate islands and still not become more than the tiniest bit genetically distinct. Anyway, so to sum up, here's some ways that you can make your birding more useful to taxonomy. Go to places that other people's don't. Try to find those areas of parapatry and take lots of media. That's really number 1. Merlin is so easy to use on your phone, and you can get pretty decent recordings a lot of the time, so easily, much more easily than it's ever been possible. And many of them are perfectly fine. Anyway, and also making sure that you're recording your media count, that you put them on sites like eBird that are going to preserve them, provide that full metadata and that permanence. Okay, I'm just going to skim over this. You can even consider media needs in your pre-trip planning. And you can let people know, taxonomists know, if you find things that seem to indicate that taxonomic change is needed. And we have lots of data now available that does make it possible to make these changes, such as the long overdue split of the chirruping nightjar in the Philippines that has this vocalization compared to all the others. And in this case, we have 3 subspecies of peewee from the Lesser Antilles and Puerto Rico that There were just very few recordings of some of them, and they seemed radically different. I went to these islands last April and got recordings, and yep, they're very different. So we're going to be dealing with that. Anyway, to sum up, in terms of ecotourism, of course, birders are super interested in taxonomic changes. We often don't pay attention to subspecies or what's recognized as subspecies because it doesn't show up on our alerts, or it's just it's not a species. But here we have a case of what's now known as a separate species, the Kalinago wren of Dominica. But until last year, it was just a subspecies of the house wren. But if you'd seen it and you'd heard it, you knew it wasn't. but anyway, so we have lots of taxonomic inertia, but we also have the increased interest in species that provides more opportunity for guides, more opportunities for protection of birds. But we also have to keep the science clean and not be driven by those considerations. And to end, we know that we are losing species. We've already lost a lot of species. Here's an example of a whole family of birds that was lost not that long ago. These videos were made not long before the total extinction of the mohoidae, the last species of moho or o'o in Hawaii in the 80s. So we do know, sadly, that our archives of specimens, photos, and sound recordings will be the only representation we have of some species, sadly. And we hope that that can be minimized as much as possible. There's still a tremendous amount to be done. We've done the consolidation of the checklist, but there's so much more to be done, and your media can make a big difference. So make it count and contribute.
Thank you so much, Pam. That was really fascinating. And as someone who is a very low-tech birder, you made a real argument for trying to use more media and attach that to my eBird checklists. We got a few questions in the comments. First, you spoke earlier about zones of intergradation, and we see that here with tufted titmice versus black-crested titmice.
Right.
And they hybridize here. And what are your thoughts about those two species? And the question was, should they be considered simply titmice with two subspecies?
Well, I would hate to see them get lumped, but on the other hand, the evidence does not seem great in terms of their species status. And the thing is that I believe that there's a proposal that is either in progress or planned to come up to the North American Checklist Committee anytime now on that. So I believe we'll see some at least decisions made soon. And again, I mean, I think the black-crested titmouse is such a special bird, but it could, I guess, if it's a subspecies, it'll just be a special subspecies. But I don't want to say which way it'll go. I don't know.
Oh, that's so interesting to learn. Some other questions that pertain to our local species here. There is a near extraliminal eastern towhee based on phenotype here using perfect spotted towhee vocalizations. What do you think is the most plausible explanation?
I don't know specifically, but of course we know they do hybridize, and probably there's some learning involved, but it could be that you said it looks like an eastern towhee, but it sounds like a spotted. Okay, so
Yes.
maybe it's a hybrid that just happens to have these combination of genes that leads it to look like a pure eastern, or maybe it happened to learn its song somehow incorrectly and near the hybrid zone or something. I really don't know. Sorry.
That's interesting. That's interesting. And it was interesting when you commented earlier that birds do have regional accents. Because I feel like I've noticed that traveling some of our common birds here, like the blue jays, sound different when I'm on the East Coast.
Yeah.
Another bird that we see a lot here this time of the year is the yellow-rumped warbler. So what's the latest on the splitting of Audubon's and myrtle?
Well, that has not been addressed recently. It has been controversial, and some of course, have advocated it. And they do have some different calls, and that's usually an indication of separate species status. The problem is that there's quite a wide hybrid zone. I believe it needs to be readdressed soon with because I believe that the latest or the proposals that NAC has already considered are some years old now, and I'm sure there's more data now that could be brought to bear. There's many things that need to be reconsidered. Let's put it that way.
Okay.
And I do want to say that anyone can submit proposals to the NAC committee. It's not exclusive by any means. So they need, of course, to meet a certain standard. They need to be scientific and accurate and all that. Yeah, we welcome proposals from non-members.
Okay.
The more, the better. I mean,
That's great to know.
Yeah.
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 You mentioned 7-point criteria for describing taxonomic relationships. Can you elaborate on those?
Oh, so this was a system that was devised by members of the BirdLife International taxonomic team, and they published a paper on it. It was also called the Tobias method or scheme, and it's basically that they had a way of scoring differences that if it totaled 7, like there were 3 major morphological differences, that would be each one of those would get 3, so that would be a sum of 9. So it would automatically be considered a species if it met that criterion. That was not met with much enthusiasm, or in fact, a lot of disenthusiasm by many scientists, and has not been widely adopted, but it has been adopted by some, and it is useful in that it provides a scheme for evaluating characters and differences, and it often does highlight those taxa that do need to be reconsidered. But for example, the WGAC committee doesn't consider, or Avalis doesn't consider 7, to be a magic number. So, it's useful, but it's not a rule.
Okay. Do you foresee a point in the future where we will need to reconsider the definitions of families, species, and subspecies into a new, more unified style of classification?
Well, even though I'm not sure I can answer that question very well, but even though species have, the definition of species has so many problems, it's actually the one taxonomic level that has at least some kind of criterion that is fairly uniformly adopted. All the others are even more nebulous. So what is a genus? Well, it's a group of species, but sometimes it's just, a genus can have just one species, or it can have 20 or 50 even. But there are taxonomists who prefer to recognize broader genera, like all of the, as an example, all of the sunbirds, almost all the sunbirds as the genus Nectarinia, or, and then there are others that prefer to have very small genera. And then there's many that are, that consider, a genus is a grouping of related species, and how narrowly you define them is an individual preference. It's really difficult to say which, it's impossible to say which is correct. It's not a matter of correctness, it's a matter of preference. So that's a source of confusion and incongruence as well. Yeah.
You talked a lot about the shift from morphology to integrative taxonomy. So I was curious that there's thousands of birds in the world. So are they each being individually studied for like DNA analysis, and that data is resulting in the taxonomy revisions? I'm just trying to kind of get a handle on how that process is being done for all the species in the world.
Right. Well, so DNA analyses, of course, are evolving dramatically and have changed so much from the earlier days. Just a couple of decades ago, mitochondrial DNA was the go-to thing that you could readily achieve without too much difficulty and without too much expense. And so we have a lot of mitochondrial DNA, phylogenies, et cetera, analyses that in many cases made, decisions were made on them. But we now know there are all kinds of issues with mitochondrial DNA that make it not exactly the most reliable source of taxonomic information. It does have utility, but it isn't foolproof by any means. So now we have genomic level phylogenies being done in a lot of cases at large scales with large numbers of taxa, but it's still nowhere near the point where we can say we have these data for everything, and but it is accelerating, and of course there's, well, I won't mention funding problems, but anyway, so the thing is though that when you design these kinds of studies, the person doing that usually has a list of taxa that they want, but in most cases, what they end up sampling isn't everything they wanted, and it isn't, and some of these samples don't work out right, can't be used reliably, and usually there's actually a lot more to it than that, because there's subspecies that should have been included, or populations that or even taxa that aren't even described yet, that people have seen or suggested might be a new species, but it's not there in the taxonomy because it's not described yet. So often, usually, the end result of a major study is still somewhat incomplete, and there's still lots of questions left over. But I don't know if I answered your question really.
Yeah, I was just trying to understand, with all the thousands of birds, how are scientists getting that level of detail to determine with precision the taxonomic order, because I noticed when the changes come out every year, one thing that happens too is birds are reordered sometimes. So just kind
more of that, because there's a major effort because there's a major phylogeny coming out that's going to be a basis for, you can expect more of that.
Yeah. Well, this was all very interesting. I learned so much. We did get a question about whether there would be a link to this recording, and this lecture has been recorded and it will live in Travis Audubon's YouTube channel. So Anne-Marie said she would stick a link to that channel in the chat. So we'll give her a second to stick it in there so you can find it there. So thank you so much. That was such an informative and interesting presentation. Really appreciated you being here with us this evening.
Well, thank you. I appreciate it.
Thank you. Hope you all have a good night. Thanks everyone for being here.
Yeah, thank you very much. It was fascinating.